tell me you're not, because otherwise I'm paying for you, which isn't going to happen. So please do come if you are SVPs, if you are coming. Otherwise, today, Grant Hughes from the Division of Rheumatology is going to present on cocaine-levamisole-associated autoimmunity, kind of unique disease that he's investigated, I think somewhat unique to Seattle, and an interesting model of how autoimmunity and drug-induced autoimmunity may develop. Grant, thanks very much. Hopefully we won't have any technical problems with the broadcast today.
All right. Thanks, Len, and Drew, for inviting me back after last year. I think I assaulted you with some basic science. I'm going to promise to be a little bit more clinical this year. This is a topic that's been of interest to me since I was a junior faculty member. I really started noticing the strange cases of apparent vasculitis or pseudovasculitis in cocaine users. And since then, we've learned quite a bit about this clinical phenomenon and also some of the immunology associated with this phenomenon. And I'm going to tell you a little story. I'm trying to connect the clinical and the basic science of what we think is going on in these patients and what it can tell us about autoimmunity. I have no disclosures. And I want to start by acknowledging Bob Wilkins. I think some of you in this room knew him well, perhaps, who passed away in 2009 after 50 years of service to UW medicine, to the UW, and 30 years as section head of rheumatology at Harborview, and I've taken over from him. But he was one of my chief clinical mentors, and I have the honor of serving professorship named for him. I'd like to thank also the lab of Keith Elkin, division head of rheumatology, and all the data I'm going to show you is generated by Christian Lude, one of the postdocs in this lab, and an internal medicine resident, Meredith Lawrence. So this is what we're going to talk about today. I'm going to talk about the epidemiology and clinical immunologic features of cocaine users with serum anti-neutrophil cytoplasmic antibodies. And this is all work done by Meredith Barnes, who actually matches one of our fellows for July, so we're very happy about that. And then some mechanisms linking a process called mitosis, which we'll talk about, to development of autoimmunity in these subjects. This is all work done by Christian Lude. So I'll roll back the clock to 2008, and this was my index case, and I think the index case at Harborview Medical Center. And I presented this case last year, I believe. A young woman with presented with acute purpura of the legs and abdomen. This is not her, but this is almost essentially what it looked like. So acute purpura with skin necrosis of the legs and trunk. She was a chronic crack cocaine user and also methamphetamine user. She had profound neutropenia upon admission. Her coagulatory parameters were normal. The investigation for vasculitis was negative, as shown above. Further investigation revealed a low titer ANA and a strikingly high titer anti-neutrophil cytoplasmic antibody level, greater than 1 to 65,000, with positive but sort of discordantly low anti-myeloperoxidase and PR3 antibodies. And the skin biopsy did not reveal vasculitis, but rather diffuse fibrin clots with some perivascular neutrophilic infiltrate. Immunofluorescence was negative. We'll come back to this in a few minutes, but I want to. This was the first of a series of patients that we encountered at Harborview. It was actually sort of the first ripple of a big wave that came through in 2009 through 2012. Come back to this in a minute. Just a brief history of cocaine in the U.S. I think a lot of us are familiar with this story. The cocaine is extracted from the coca plant, which was used medicinally in South America for centuries, purified and used as a sort of over-the-counter medicine in the U.S. and other countries for a long time, and then really became a drug of abuse in the 1980s, almost all through this one man here, Pablo Escobar, who was in the early 1990s, one of the 10 richest men in the world through exporting and selling cocaine in the U.S. And then in the 1990s, the freebase form called crack cocaine became almost epidemic in certain urban centers in the U.S. It remains a fairly large public health issue with over 1.5 million active users as of 2014 through a variety of methods of use and exposure, including snorted powder cocaine, smoked crack cocaine, and IV injection of solubilized powder cocaine hydrochloride. And interestingly, and this may have some relevance in what we're going to talk about, crack cocaine use seems to be concentrated in older cocaine users, whereas which may have some bearing on what we'll talk about. Okay. I do want to talk a little bit about allergy. There are a variety of cocaine-associated syndromes. As we know, it's a powerful CNS stimulant with powerful sympathomimetic effects, so it's cardiovascular and CNS effects, but also has a lot of rheumatologic and some manifestations that mimic allergy. I think a lot of you are familiar with this syndrome of cocaine-induced midline destructive lesions associated with snorted cocaine use. Patients present with sinusitis, epistaxis, and nasal obstruction. And it can mimic what we call limited Wegener's granulomatosis, but it doesn't have the extra airway manifestations. And there's no granulomas or vasculitis and biopsy. And these patients have very high titer ANK. And this was first described back in the early 1990s, and that was really the first link between cocaine exposure and anti-neutrophil cytoplasmic antibodies. I'm not going to talk too much about this, but I do want to just remind you how anti-neutrophil antibodies are tested and are detected in the lab. It's hard to see this, but basically you're detecting circulating serum IgG antibodies that recognize cytoplasmic antigens and neutrophils. And the way the healthy neutrophils or neutrophils from healthy donors are fixed in formalin and ethanol. And this causes actually relocation of cytoplasmic granules such that highly cationic molecules such as neutrophil elastase and myeloperoxidase actually migrate to the perinuclear area, whereas the other granule contents stay put. And this is actually the basis of these different patterns of C-ANCA and P-ANCA. These are actually artifacts of the fixation process. And in the diagnosis of systemic vasculitis, these patterns, P-ANCA and C-ANCA, are associated with antibodies against myeloperoxidase and proteinase 3. And these combinations of specific immunofluorescence patterns and specific autoreactivities are what are specific for the diagnosis of systemic vasculitis. And importantly, in patients with biopsy-proven vasculitis, they'll have either one or the other, but not both of these reactivities. So these are somewhat mutually exclusive and actually have different genetic associations. Why is that important? Well, we'll talk about that in just a second. So the ANCAs in the cocaine-induced midline destructive lesions appear to be very different than in idiopathic vasculitis. They're very high titer P-ANCA with variable levels of MPO and PR3. And so some early studies asked, well, what's the reactivity in this ANCA? And it turns out that a lot of the reactivity is against neutrophil elastase, another cationic molecule in neutrophils. And this is just some data from 2004 showing CINVL on the left having very high reactivity against neutrophil elastase, whereas Wegener's granulomatosis, microscopic polyangiitis, and some disease controls really have low-level reactivity. Keep the year in mind. We'll come back to this. This is from 2004. Okay, so let's come back to our index case. This is the young woman, crack cocaine user with thrombotic purpura of the skin, profound neutropenia, and really outrageously high ANCA titers in the serum. So this, as I mentioned before, was, at least at Harborview, was the initial index case of a wave of cases that began around 2008. And since then, there's been dozens of publications describing this in other urban centers in the U.S., New York, Boston, San Francisco in particular, describing some common clinical manifestations in these cocaine users. Arthralgia, retiform purpura, which is that necrotic skin lesions that I just showed you, and neutropenia. So here's another picture of somebody with retiform purpura of the legs. And these clinical manifestations are associated with acute drug exposure. I'll come back to that at the end. So in other words, if a patient abstains from cocaine use, they tend not to get into problems. When they relapse, these things can come back, and then they go away if they stop using drug again. So there's an association with acute drug exposure. It seems to be associated with multiple forms of routes of cocaine exposure, so both injected and inhaled. And occasionally associated with features traditionally associated with ANCA vasculitis, such as posse-immune glomerulonephritis, immune complex glomerulonephritis, and even pulmonary hemorrhage. But these are uncommon presentations. What about the immunology in these patients? So exceptionally high ANCA titers, as I mentioned. They're usually P-ANCA. So if you look at the literature, most cases describe P-ANCA reactivity with variable MPL and PR3. And when studied, anti-neutrophil elastase antibodies are almost universal. So, interestingly, these patients have anti-cardiolipin antibodies, and they're almost always IgM. And anti-ANA and double-stranded DNA antibodies are occasionally reported, but don't appear to be a major feature of this syndrome. Now, the correlation of these immunologic parameters with clinical symptoms has really not been well described, and that's what I'm going to talk to you about in a minute. So, well, what's going on? Why did this show up in 2008, 2009? Well, in 2009, the DA identifies levamisole as a common cocaine adulterant, present in over 70% of seized cocaine in 2009, whereas in 2007 it wasn't in there. It's an levamisole is an immunomodulatory drug that's been used since the 70s for trials for RA, pediatric nephrotic syndrome. Is actually an anti-helminthic drug used in veterinary medicine still today. But this drug was taken off the U.S. market in 2000 due to toxicity. What was that toxicity? Neutropenia, rare necrotic skin lesions, particularly the ears and face, and in some case reports developed in the bank. Sounds familiar, doesn't it? Case solved? Yeah, question.
Is there any reason why they started using levamisole? Did anybody ever postulate that?
Yeah, so it's unknown, and there was one of the local Seattle papers that did a huge expose on this in 2010 that explored this in detail, and they really couldn't come up with a conclusive answer, but some ideas is that it actually enhances the CNS effects of cocaine. It doesn't interfere with street test for purity or taste, so it doesn't affect the physical properties of the cocaine, and so made for a better product. It probably wasn't put in to cut it to reduce cost of production because the actual levamisole is somewhat expensive. So those are the current ideas. So, I think a lot of people said, okay, case solved, and started calling this syndrome levamisole-induced vasculitis or vasculopathy, because it made sense to blame levamisole as a cause of neutropenia and purpura. But as I just showed you, cocaine use before the addition of levamisole was clearly associated with loss of tolerance against nuclear antigens and neutrophil elastase in particular. And renal and pulmonary manifestations have not been described in clinical trials of people using levamisole. So there's something else going on. This is not just levamisole. So, skip over this. There haven't been a lot of trials actually looking at this. There've been, if you look at the literature, it's riddled with small case reports, 1 patient, 2 patient, and a few case series. There's one of 18 subjects, and there's a more recent one actually of 30 subjects from MGH. But there's been little attempt to correlate the immunology with the clinical manifestations. So this is where Meredith stepped in. She's really interested in doing a research project that was related to immunology and rheumatology. And so we had a small series of patients that have been referred to rheumatology for evaluation of vasculitis who were cocaine users with high titer ANCA. And we looked at those subjects and made some initial observations. They appear to be almost all Caucasian, female, and between the ages of 40 and 50, which was interesting. hepatitis C virus infection was prevalent. I think it was about 80% of these subjects. While purpura and skin manifestations, including ulcers and neutropenia, were quite common, there was also reports of glomerulonephritis and pulmonary hemorrhage. And they had universally high titer P-ANCA with variable MPO and PR3 reactivity, as had been reported. And interestingly, the ANCA appeared to be persistent in these subjects. So based on these initial observations, which Meredith presented at our national conference about a year ago, she formulated some hypotheses. One, she hypothesized that this cocaine-levamisole associated autoimmunity occurs in a demographic subset of cocaine users. The patients we were seeing didn't seem to fit what we assumed was the general cocaine using population. And by the way, this cocaine-levamisole associated autoimmunity term is something that Meredith and I came up with as I think, a better descriptor of the syndrome than levamisole-induced vasculitis or levamisole-induced disease, just because we don't know which of these drugs is causing it. Vasculitis is not always a universal feature in these patients. So another one, we saw a lot of hepatitis C infection. We hypothesized that it was associated with hep C infection. Even though hep C is common in the cocaine population, we hypothesized this. And again, feel free to interrupt me at any time with questions if you have. We thought that because thrombotic purpura was a major manifestation here, we thought it might be associated with the antiphospholipid antibodies that I mentioned earlier, which you may know are associated with thrombotic disease, with venous and arterial. And we thought, because the renal and pulmonary manifestations were uncommon, that they might be associated with PR3 and or NPO reactivity, which we know is associated with idiopathic vasculitis and these clinical manifestations in non-cocaine users. So these were some of our main hypotheses.
Has this been seen in other countries, or is this unique in the U.S.?
It's been reported in Britain, I know, in association with levamisole that's found in the cocaine there. But it's almost all of the reports have come out of the United States.
Does the cocaine come into this country already cut with levamisole?
Yes.
Yeah,
it's added at the source.
In your index case, you said that there were no coagulation abnormalities. Is that what you think with antiphospholipid antibodies being positive and things like that you might see coagulation cascade problems?
Yeah, so that's a good point. Sometimes in patients with antiphospholipid antibodies, you'll see prolonged PTT or sometimes even INR, but not all of them. In this particular patient, we didn't see that, but I will say that in others, we have seen prolonged PTT associated with their antiphospholipid.
Has anybody looked at the amount of levamisole in the cocaine that these people are using? Is there any differences between, like, is it more levamisole to cocaine? These people who tend to get it?
No, that hasn't been looked at. I mean, there are a couple studies that look at the amount, but it hasn't been studied extensively that I'm aware of.
Levamisole is still a licensed drug in other countries?
Yes, I think it is. And it's a veterinary medicine. So what Meredith did is just simply looked at our large database from 1990 to 2015 and said, I want to find everybody who's had a positive cocaine urine test and positive ANCA. And that was the inclusion criteria. And she excluded anybody with the biopsy-proven ANCA vasculitis or any drugs that we know that can induce ANCA vasculitis, such as propylthiouracil or hydralazine or minocycline. She did chart review, and we also compared to the background populations of cocaine users and those with positive ANCAs using something called the De-Identified Clinical Data Repository, which, if you haven't heard about this, you might want to just get on the ITHS website and take a look at it. It's pretty, if you're UW affiliated, you should have access to it. So one of the things we observed is that using the DCDRs that, this is just 2010 for present, that a significant portion of those being tested for ANCA who have a positive result are actually cocaine users. It's like 11%, which I thought was surprisingly high. So cocaine use may be a major risk factor, maybe actually a fairly common cause of drug-induced autoimmunity. We just don't know. Okay, so here's our cases. We identified 52 were included. One was excluded due to diagnosis of vasculitis, leaving us with 51. And you can see after about 2008 or so, the number of cases detected by our screen really rose. And keep in mind, this is a biased population. So who do you check for cocaine use? Well, people who say they're using it or people whom you suspect are using it. Who do you check ANCAs in? People whom you think have vasculitis, presenting with certain clinical manifestations. So keep in mind, this is a highly biased population and represents probably a sliver, we think, of cocaine users who may have ANCA reactivity. Looking by titer, so on the left, on the y-axis is 1 over the ANCA titer, so the higher the number, the higher the titer. You can see that after about 2008 or so, there's a cluster of extremely high titer ANCA levels in our subjects. We actually think that ANCA titer can be a discriminating factor in determining whether it's idiopathic ANCA or drug-induced. And we're actually working on a separate project to look at diagnostic utility in titers. So let's just look a little bit on the immunology of these patients. Surprisingly, we found a lot of atypical ANCA patterns, which has not been described in the literature. And only about 60% of our subjects had the classic or well-described P-ANCA pattern. And what was also interesting is that, like others, we found patients with both PR3 and MPO reactivity, but we also found a significant portion of people with double reactivity, which, as I mentioned before, is not seen in idiopathic vasculitis. And if you actually compare that to ANCA-positive individuals that we found using the DCDR, which was only about 310 actually, the cocaine population appears to be significantly enriched in double positivity. This may also have some diagnostic utility in discriminating idiopathic ANCA from drug-induced ANCA disease. What about the demographics? So, in our series of 50, in our population of 51, they were indeed predominantly female, predominantly Caucasian, and with an average age of 46, so consistent with our initial observations. So we went and pulled demographic data from anybody who had a positive cocaine test within our medical record system at 6,200, and indeed our cocaine-using population with ANCA appeared to be enriched for female individuals, those of Caucasian ancestry, and actually African American ancestry appeared to be somewhat protective in this population, or underrepresented in this population. So our population appears to be more female, less African American than what we deemed the background cocaine using population, suggesting both sexual and genetic factors. Here we are looking at anticardiolipin positivity. So of those tested for anticardiolipin antibodies, the majority were positive in our population, whereas in the general cocaine population it was less frequent. And we also discovered that lupus inhibitor, which goes along with antiphospholipid antibodies, was significant, was very common in our population, much higher than in the cocaine-using population tested for lupus. I'm not going to go too much into the clinical manifestations for the sake of time. Better for another talk, but some of the things we observed were frequent hematologic abnormalities, which makes sense. About 60% of our subjects had neutropenia, but a lot of them had anemia, and very few had thrombocytopenia. So there's some other things going on. Very commonly observed renal abnormalities, and this was defined as proteinuria or abnormal creatinine. So we don't know exactly what's going on. Only a few of our subjects had glomerulonephritis, as has been reported in the literature. And I think what's interesting is only about 60% of our subjects had any dermatologic manifestations, which, if you look at the literature, this is a hallmark of this syndrome. So, in other words, we're finding cocaine users with ANCA who don't have these classic manifestations. They were tested for ANCA probably for other reasons, such as renal disease, or just because of awareness of this syndrome. So, I think these data are suggesting that the clinical problems associated with ANCA and cocaine users are probably more diverse, and there's probably a subset of patients out there with ANCA who don't have any disease also suggests that ANCA alone is not pathogenic in the syndrome. We're going to come back to that in the last 20 minutes.
What about the hepatitis C? That wasn't up there.
Oh, yeah,
Was
sorry.
something-
No, it was, Nick. Thanks for bringing that up. So we didn't find any enrichment of hepatitis C, either exposure, history of exposure, or viremia in this group. So it doesn't appear to be a risk factor, and it appears to be a feature of cocaine use, which makes sense in terms of risk behavior. So we were doing some logistic regression analysis to see which of these factors independently associate with various clinical manifestations. And this is work in progress. I can only show you some initial findings. The skin manifestations in these patients, so either purpura or ulcers, seems to be associated fairly strongly with female sex, or as we hypothesize, anticardiolipin antibodies. We're still getting the data on lupus inhibitor, but we think it's going to be the same. So this suggests that these antibodies could be playing a role in the pathogenesis of purpura. The MSK manifestations were also associated with female sex. Not a huge risk factor. Not associated with any of the clinical manifestations that I showed you earlier are age, race, PR3 or MPO, or hepatitis C virus. So we don't think that these PR3 and MPO antibodies, as we initially hypothesized, are driving, let's say, nephritis or pulmonary hemorrhage. And we don't think that the hep C co-infection is driving disease either. We think something else, anticardiolipin antibodies or something else, are driving disease. And so this is just a summary of what Meredith found. In cocaine users, female sex and race seem to, or probably genetic factors seem to influence the development of ANCA, or at least ANCA prompting, or symptoms prompting an ANCA test. But we also found quite significant enrichment for lupus inhibitor, double positive ANCA.
Is that what do you mean by lupus inhibitor?
So that's the phospholipid dependent inhibitor of the PTT in vitro.
Okay.
That's associated with antiphospholipid antibodies. It's actually the strongest predictor of thrombotic disease in patients with antiphospholipid syndrome. So we think it's clinically important. We just need to look at it quite a bit more closely. So some of the future questions we have on this project are, what's the true prevalence of ANCA? How common is cocaine-induced drug autoimmunity? It may be one of the more common forms of drug-induced autoimmunity in this country. And what are the clinical correlates of this lupus inhibitor? So we need to go back and look at that. Does the form or routes of administration matter? So far, we don't have a hint that is the case. And what are the relative roles of cocaine versus levamisole in both developing ANCA and in the various clinical manifestations associated with it? And as I hinted at before, can patterns and titers of ANCA really help discriminate drug-induced autoimmunity from idiopathic autoimmunity? And actually, should anti-neutrophil elastase be part of our ANCA panel? So Mark Wenner, head of laboratory medicine now, has been very interested in this and considering adding it to our panel, particularly in our urban population that we serve. I'm going to change gears here and get basic on you. So any questions on the clinical data?
So on HLA haplotype, is there any times to look at that?
We don't know. I mean, in idiopathic pancreatitis, there's certainly HLA associations. But these data suggest that if we look, we're going to find some HLA associations in our cocaine or CLAA population.
Have you thought about looking at the prevalence of ANCA in a drug treatment population, like just cocaine abusers? Because you have the other, you have the prevalence of cocaine use in ANCA positive.
Yeah. So, that's something we thought of doing. So to assess the true
Right.
rate.
How often?
Yeah, one of the ways we can do this is capture a lot of subjects with the Harvard U ER. So we want to develop a prospective trial where we just capture any cocaine user and just check their serum. Just get a better estimate of what the prevalence is. Because I bet it's a lot higher.
I think one of your slides seemed to show that the numbers of patients were actually almost like dipping down. Is anything different going on as far as cutting cocaine?
So that's a great observation. Yeah, so I've looked into that. I don't have any more recent data on the percentage of seized cocaine that has levamisole, but you might predict that it's actually dropping based on those numbers. or people just aren't checking in as much as they did. So there could be an awareness thing as well.
Is this unique to cocaine or any other illicit drugs?
Not other illicit drugs that we know. So the prescription drugs, propyluracil, minocycline, and hydralazine can induce ANCA, that's P-ANCA and anti-neutrophil elastase specific, but those are actually associated with frank vasculitis, glomerulonephritis, pulmonary hemorrhage, etc. But not other illicits that I know of. Okay, so, so we wanted to ask, what are some of the mechanisms linking cocaine exposure and ANCA to these clinical manifestations? And, how might cocaine and or levamisole directly induce these manifestations? We know they're linked, particularly levamisole. And then how does exposure to drug lead to loss of tolerance, particularly to neutrophil cytoplasmic antigens like neutrophil elastase? And finally, are ANCA pathogenic in these subjects? I just told you that patients can have ANCA and not have any symptoms or clinical manifestations. So we don't think that they're sufficient to be pathogenic, but can they be pathogenic in certain situations? So some of you may recognize this issue of Science from 2004 that identified a newly recognized form of neutrophil activation called mitosis, or formation of neutrophil extracellular traps. I'll just say right now, we think that this phenomenon could be linked to all three of those questions that I just showed you. So neutrophil extracellular traps are a result of a form of activation-induced programmed cell death in neutrophils and other granulocytes. So there are actually other forms of extracellular traps, but neutrophils appear to be the dominant source and the most common, they're the best described cell set that makes nets. And so various forms of activation can induce nets, including chemokines and cytokines, particularly TNF-alpha, toll-like receptor ligands like LPS, antibodies, and we'll talk a little bit about that in a second, and immune complexes can all induce mitosis. And so what is it sort of at the cellular level? So the neutrophil is triggered to extrude its DNA, chromatin, sometimes mitochondrial DNA, depending on the stimulus. And it's laced with neutrophil enzymes. So neutrophil elastase appears to be the dominant enzymatic component of these nets, but also PR3, MPO, cathepsin G, citrullimated histone 3, and a variety of other components. What are the function of these nets? Well, in this picture in the lower right, you see nets sort of trapping some bacilli. And the idea is that these nets have antimicrobial activity. So they physically trap microbes and then bring them in close proximity to antimicrobial products, such as neutrophil elastase, PR3, etc. They also have prothrombotic activity. So if formed in tissues and intravascularly, they really promote thrombosis. And some forms of nets appear to be inflammatory. That's going to be important in just a minute. I just want to highlight some key intracellular processes that are required for netosis because it relates to the data I'm about to show you. These are intracellular processes. So reactive oxygen species production is required to form nets, either NADPH derived, which we're all familiar with, most of you are familiar with, or mitochondrial derived reactive oxygen species. The release of neutrophil elastase and myeloperoxidase from granules into the cytoplasm is required to break down the nuclear envelope and to decondense the chromatin. And interestingly, citrullination, so this modification of arginine to citrulline, an enzyme called PAD4, plays an important role in this whole process. It probably has to do with histone modification and conformational changes, but it's not really well understood. All of these things are required to release these traps into the extracellular space.
This may be a naive question because I don't know anything about this, but is this an active process, another mechanism by which neutrophils play a role in host defense, or
Yes.
is this just apoptosis, a different name?
No, this is an active, programmed form of cell death that is important in host defense and also tissue injury in a variety of autoimmune and inflammatory conditions.
So the usual way you think of neutrophils killing bugs is they phagocytose them and kill them with MPO and hydrogen peroxide intracellular. This is yet another killing mechanism.
This is, thought to be an extracellular killing mechanism. So these nets are now thought to be involved in idiopathic ANCA-associated vasculitis. And this is a busy slide, but I'll want to point out a couple things. So, a couple things that illustrate how ANCA can be pathogenic through induction of mitosis. So, in this image, you see a neutrophil being primed by soluble mediators here, C5a and a cytokine, tumor necrosis factor alpha, which activates the neutrophils to externalize some granule contents. Those granule contents are the target of ANCA in both idiopathic and cocaine disease. So the idea is that the antibodies recognize that and also co-engage Fc receptor 3 alpha on neutrophils. And that engagement of Fc receptors actually pushes forward neutrophil activation and drives netosis. And those nets can be released in tissues or in circulation where they can induce tissue injury through, if they're inflammatory, or thrombosis. And so this idea that ANCA can become pathogenic in certain situations of neutrophil activation is one in a series of pieces of evidence that suggest how these antibodies can cause disease in idiopathic vasculitis. So we wanted to ask, are these mechanisms active in cocaine-associated autoimmunity?
Is there any evidence that those nets are clearing the antibodies that are there? And that they're just acting as a sieve or something to clear the antibodies out of this situation?
Well, I think they definitely would bind up antibodies, but the nets themselves appear to be what cause damage in certain situations, in the kidney, in the lungs, and maybe thrombotic disease in arteries and veins. Yeah. So Christian Lude and Keith Elkin's lab and I got together and said, this netosis thing could be involved in all of these processes of cocaine-induced autoimmunity. So we asked some simple questions, which are listed here. I'm going to show you the data here. So we asked, the first thing is, can these drugs induce netosis in vitro? And these are some pictures that say yes, both of these drugs, cocaine and levamisole, at physiologically relevant concentrations, which I'm not showing you, can induce netosis. So what you're seeing in the upper left are healthy neutrophils without stimulation. DNA is blue, and neutrophil elastase, which, as I said, is a dominant enzymatic component of NETs, is a nice surrogate to measure NETs. And as positive control, we use PMA, which drives this NADPH oxidase dependent pathway of netosis quite aggressively. It's a very potent stimulator. You can see the release of DNA laced with neutrophil elastase. And interestingly, cocaine and levamisole do the same thing, but not. So it looks very different. So you see some neutrophils that appear intact, and others that are released. You can't see the blue very well, but blue DNA just laced with lots of neutrophil elastase. And one of the ways you can quantify this is actually to measure the DNA that's been released from the nets. And I'm not going to go into the details much, but I just want to show you the data of how much DNA is released. So PMA on the left, figure B, induces a huge amount of DNA release, whereas cocaine and levamisole induce a lesser amount. But if you look at the quality of these nets, they appear to be enriched in neutrophil elastase. And the images I showed you suggested that. So if you quantify that on the right, if you just, or figure C and D, compare the ratio of neutrophil elastase to DNA, you see that these drugs induce nets that are unique, or at least different from PMA, and they're enriched in neutrophil elastase, which is a dominant target autoantigen in cocaine-induced autoimmunity, or associated autoimmunity.
Do cocaine and levamisole stimulate a traditional oxidative burst as well?
We don't know. We haven't looked at that, but I'll show you some indirect data about inhibition of NADPH oxidase and the relevance to this process.
Do you have any data on cocaine and levamisole together?
We do. It's sort of a work in progress. It's an important question about synergy. I I'm not going to show you any data, but that's something we're really looking at.
And is there a dose response with cocaine or levamisole? Is it
Yeah.
like
Yeah, so there's a dose response, and we chose a dose that was within the physiologic range reported in people using levamisole, clinical trials, and cocaine users that induced a significant amount. So I'm not showing you those data, but there's a clear dose response here. So drilling a little bit more detail on pathways of mitosis. There are three major pathways that are appreciated. One is the NADPH ROS pathway, which I mentioned before. Rapid neutrophil death. This is mostly nuclear DNA chromatin, and PMA triggers this pathway. There's something called vital mitosis, which upon neutrophil exposure to certain gram-positive bacteria and some other stimuli, they release smaller nets that are actually mostly mitochondrial DNA, and it's driven by mitochondrial ROS. And these neutrophils can continue to phagocytose and migrate even after they've released their DNA. It's a very interesting pathway. And then the most recently described one was actually described by Christian Lude, Keith Elkin, Mariana Kaplan, NIH, just published in Nature Medicine. Is an immune complex-induced mitosis, which seems to be unique in that, like vital mitosis, it requires mitochondrial ROS, and it's enriched for mitochondrial DNA. But these nets are very inflammatory. So when monocytes see them or other immune cells see them, they release inflammatory cytokines, including traditional inflammatory mediators like IL-6, but also type 1 interferons. And in vivo, they're immunogenic. So they drive antibody production against components of the nets. And that's a very interesting observation. I'll show you why that's important. So one of the things we asked was, which type of mitosis are these drugs inducing? So one of the ways you can do that is block these various pathways of citronellation and reactive oxygen species. So we just did a simple experiment in vitro. Actually, Christian did. I didn't do this experiment. Blocking PMA-induced mitosis. So on the left, there's no inhibitor. Everything's normalized to 1. And you can see with, at the bottom, inhibiting these various components of the net, intracellular components of the mitosis. You can see that if you block NADPH oxidase with apocyanin, it almost completely knocks down mitosis induced by PMA, which is it's been reported. But if you block mitochondrial ROS on the far right, it doesn't do anything to the PMA-induced mitosis. And this is all known. But our drug-induced mitosis seems to rely on both pathways. So it's inhibited by inhibitors of NADPH oxidase and mitochondrial ROS. So it has features of both of those first two pathways that I mentioned. Interestingly, if you quantify the amount of mitochondrial 16S to chromosomal 18S DNA, these nets appear to be enriched in mitochondrial DNA, suggesting they may have some of these inflammatory features that mitochondrial, or immune complex and/or vital mitosis. So not only are they enriched in the autoantigen neutrophil elastase, they're enriched in mitochondrial DNA. So they're it appears to be a unique form of mitosis. Finally, some of you may know that neutrophils are a major cellular source of the B-cell survival differentiation factor, BAFF. So we asked, do these drugs induce the release of BAFF? And this is some preliminary data suggesting that, yes, these drugs can enhance the release of BAFF from neutrophils. This could just be from cell death. This is actually a long-term culture. But it does suggest that these drugs can induce a situation where you have a release of autoantigen, neutrophil elastase, in the setting of potentially inflammatory neutrophil nets in the presence of the B-cell survival factor. So all these things really suggest a way in which drug-induced nets could contribute to loss of tolerance against neutrophil antigens in the right genetically susceptible individual, for example.
Is this also the mechanism by which more traditional drugs cause autoimmunity? Is that where you're going?
Yeah, well, there's not a lot known, but it's a great question. So there's only one paper looking at this in vivo, and it's with propylthiouracil, which can induce nets in vitro, and those nets are unique in that they resist degradation by DNAs. And so if you take those nets and inject them into rats, they develop anti-myeloperoxidase antibodies from glomerulonephritis. So it's the one study we have that shows that drug-induced nets can have unique properties that are immunogenic or inflammatory and can actually contribute to loss of tolerance. So we're hypothesizing that the same thing may be going on here, and we're working towards some in vivo model. That's a great question. So one of the other questions we had is, we know that in these patients that their antibodies recognize neutrophil elastase, so we just wanted to see if the antibodies recognized net-associated antigens. And what you're looking at here in the top row are neutrophils that have not been stimulated, and in the bottom row, those have been activated by levamisole, the exact same conditions I showed you earlier that induce mitosis. And we've incubated them with 3 different purified immunoglobulins: healthy control immunoglobulin, AC, which stands for ANCA control immunoglobulin, which are people with not cocaine users who have high titer ANCA. So they're high titer ANCAs and non-cocaine users, and then our CLAA subjects. And what you can see in the top row, if you just look at the inset, is as expected, both the ANCA control and the CLAA IgG recognize neutrophil cytoplasmic antigens by definition. It's expected. And then upon mitosis, the CLAA IgG, but not really the ANCA control, strongly bind to neutrophil-associated antigens. And we think this is neutrophil elastase. We've, in a series of subjects, just quantified neutrophil elastase reactivity, and we're doing this in other subjects. They clearly have high levels of reactivity as compared to non-cocaine agents. So what we've shown so far is that either drug at physiologically relevant concentrations can induce mitosis, which could contribute to a variety of these phenotypes, particularly the purpura and the occasional cases of vasculitis, maybe renal disease. So it's a potential mechanism linking this direct drug exposure to disease activity. And we also found some mechanisms by which those drug-induced nets could help break tolerance to neutrophil antigens in ANCA. So our next question was, like in idiopathic ANCA vasculitis, can those antibodies be pathogenic through promotion of mitosis? So once someone develops an antibody, can they actually promote further mitosis? And the answer, at least in vitro, appears to be yes. So this is where we've taken neutrophils and stimulated them in the same, stimulated them, they're pre-treated with, if you look in the bottom, pre-treated with IgG, the same IgG subsets that I showed you in the last figure, and then pulsed with drug and measured the net release, just like I showed you before. And what you can see here is that on the far right, PMA, which is a potent inducer of DNA release, is not really affected. If you look at the y-axis, it's much higher than the other two. However, in the presence of ANCA, drug-induced DNA release is markedly enhanced. And this doesn't appear to be specific to CLAA IgG, but appears to be more robust in that population. So the important point here is that the presence of ANCA can markedly enhance, triple in some cases, drug-induced mitosis. So this may be a mechanism linking presence of ANCA to clinical manifestations with acute drug exposure. Because remember I mentioned that ANCA alone is insufficient. So this may be one mechanism linking those 3 things together. So I want to wrap up here, summarize this pathogenesis model that we're working on. Is either of these drugs can induce mitosis, and we don't know if they synergize yet. We're looking at that still. They can induce mitosis, which could play an important role in causing some of these clinical manifestations, particularly the thrombotic skin disease, perhaps vasculitis. Importantly, these unique drug-induced nets, by virtue of enrichment for target autoantigen, potential inflammatory characteristics, and co-release of BAFF could, in a genetically susceptible individual, lead to loss of tolerance to neutrophil antigens, which, once established, upon subsequent drug exposure, enhances the same process and probably lowers the threshold for clinical symptoms. This is precisely what we see in our patients who have used cocaine for years and have persistent high titer ANCAs, and they're fine as long as they don't use the drug. As soon as they use the drug again, this happens. So we think we've sort of hit on some potential mechanisms linking cocaine exposure to loss of tolerance to clinical manifestations. And we have a lot of future questions. So the synergy question is top on our list. Also, we want to know, what intracellular pathways are really involved. And there's a competing group looking at this that's actually identified muscarinic receptors on neutrophils as the point of interaction with levamisole. So another group is looking at this too. So levamisole binds to M3 receptors on neutrophils and triggers mitosis through AKT ERK dependent pathway. It's very interesting. We also want to know what the effects of these nets are on T cells and B cells in vitro, and on monocytes as well. So are they inflammatory, as we think they are? Do they promote B cell activation, T cell activation? Because one of the things interesting about these ANCA titers is that they're IgG and they're persistent. So this suggests long-lived B cell class switch recombination responses, which don't necessarily need T cells, but it suggests that T cells are involved. And this fits with the HLA class 2 association with idiopathic vasculitis. And we'll probably find them in this syndrome too if we look. And then finally, we really want to go in vivo. So our drug-induced nets, like they are with PTU-induced nets, are they immunogenic in vivo? We're exploring ways that we want to do that. So I'm just going to wrap things up here, and then maybe have a little bit of time for questions. So cocaine and levamisole-associated autoimmunity could be a relatively common form of drug-induced immunity. It appears to occur in a subset of cocaine users, suggesting genetic and sex-dependent factors. And the drug-induced net may be an important source of autoantigen in this group. And then the CLAA-associated ANCA may be pathogenic during acute drug exposure, providing a mechanistic link to clinical manifestations in people who developed autoimmunity previously.
Is netosis seen in all neutrophilic activation syndromes?
No.
Why is it unique in this? Why isn't it seen in all neutrophil activation?
Right, so there's certain triggers that, so this is a program form of neutrophil cell death, so it's linked to certain triggers. So if you expose neutrophils to IL-8, for example, they may not induce, you may not get, actually I think IL-8 is an inducer, but there's certain activators of neutrophil activity that don't induce netosis, so they induce phagocytosis without netosis. So it's not a universal.
Like pneumonia, is it just seen in the tissues of pneumonia of all patients? Is this just important if it's in the intravascular compartment? Is it, in tissues that maybe it's not important?
That's a good question. I don't know the answer to that. But it's clear that the location of the netosis could have a major impact on the pathogenicity.
Does this happen in DIC?
I think there's some evidence that it does occur, and that's what some of the evidence based on its prothrombotic effects. But I don't know that literature very well.
So I see that cocaine and levamisole have very similar kind of effects in your in vitro model. Have you looked at structure of them to see if there's any, if there's certain parts of it, and have you looked at analogs that have similar structures to see if they also do the same type of action?
No, we haven't. I mean, I think I showed you some pictures of the chemical structures. There could be some similarities, but we don't even know if they bind the same receptor, like the muscarinic receptors. But that would be a place to look. Do cocaine and levamisole both bind to M3 receptors? So that would be consistent with their similar in vitro effects. I know we haven't done any of that.
And just, in inner city asthma, inhaled cocaine is thought to be an important stimulus of asthma. And your discussion of the ANCA is just making me think about, EGPA or Church-Strauss syndrome, which is kind of frustrating because when we order ANCAs in that syndrome, of course, they're only positive in about half of the patients. And it seems like the problem is that you're not looking at the right antigen. So your discussion there, eosinophils, of course, form nets as well.
Yeah.
And they have a different set of, oxidative enzymes that are at play and so forth. I don't know if you had any thoughts about that. It seems like that's something that needs to be looked into more with that syndrome.
Yeah, well, I think, those are great observations. I would say, though, that, we don't know the extent of cocaine-induced autoimmunity. We're just looking at something that's been measured, which is ANCA. But there's clearly other reactivities that are induced, antiphospholipid, lupus inhibitors, and there could be other broader activities that are important for eosinophil activation, Th2 immunity. We just don't know.
Interesting, yeah.
When you were talking about the differences between those enriched in mitochondrial DNA and those enriched in chromatin DNA, are there certain inflammatory mediators that are known to be associated with one or the other? Have that been the differences between the two been characterized?
You mean in terms of induction?
No, like when you were talking about things that are bound to the DNA that actually come out into the net, are there different things bound to mitochondrial DNA versus chromatin?
Yes. So, and I can't speak with too much authority on this, but the protein components of those nets are very depending on the stimulus. And some of those protein components are actually contribute to triggering inflammation. So, for example, LL37, I think it's a protein component that helps drive interferon production in certain cells in plasmacytoid disease. So the protein constituents of the net can really determine their inflammatory capacity.
If you look at the thrombus formations in the vessels that are thrombosed, are these complexes seen in them when you stain for those?
Nobody's looked. So the data I just showed you is unpublished, and there's only one other group that I'm aware of that's actually looking at mitosis and drug-induced autoimmunity, cocaine-induced autoimmunity. So I don't think I'm sure there's somebody who's looking at it now.
In your lab, probably.
Where do you get the cocaine for the in vitro experiment?
There's a lot of regulations.
Have you had a chance to look at the affected patients just for any genetic markers that are traditionally associated with autoimmune risk?
Like B27?
Yeah, just curious.
No, we haven't. It's something we want to do. We just collect just get some triple blood samples and just gene
Any potential role for mast cells? I have to ask, as an allergist. Mainly because opiates are non-specific mast cell releasers. They activate mast cells in a non-atopic way.
I
It'd be interesting.
It hasn't been. That it actually is not what this is in our setting. So we haven't looked at hard for it. We should.
Pretty neat that a clinical observation turns into a 4-year cycle there. Yeah. I mean, that's
I might actually